So I'm back, at least in body, if not in spirit. You'll notice I'm drinking my coffee here as well. I was 9 hours time difference, and so now I'm about 4.5 hours time difference probably right now. Dr. Bartlett, I spoke with him briefly before class today. He said there were a couple of slides that you didn't get to, and that he didn't do the clicker questions, even though one of the clicker questions had absolutely nothing to do with anything that any of us had talked about yet. We may get back and talk about superquelling a little bit later on. So, but I did come up with a couple of clicker questions for his lecture, and the first one of those is as follows. The main function of alternative histone Sen-PA is in telomere maintenance, centromere formation, chromosome segregation, histone code reader interactions, histone fold interactions. And again, feel free to discuss. I'm sure it was one of those things he didn't cover at all. Yeah. Ten, 5, okay. One of the nice things about clicker questions is it gives me a chance to drink some more coffee, which is very good. So again, main function of alternative histone Sen, which stands for what? CN? Centromere, yes. So that should give you a very good idea. But also this reminds me that I think I didn't do a very good job of talking about centromeres and telomeres and so on and so forth earlier on. So here, telomeres, where do you find telomeres? The ends of chromosomes. Do you need special histones for telomeres? Actually, it turns out you do, but we haven't talked about them. And it's certainly not Sen-PA. Centromeres are what you have in the middle, i.e. really not at the ends of chromosomes. Why do you have centromeres? What are they there for? So to get the separation, really, of chromosomes, they have some way of the microtubules interacting with them. So it has to do with chromosome segregation, pulling those things apart. Histone code readers, that's all about what? The modifications that, again, I'm sure Dr. Bartlett didn't talk about, so it shouldn't be on the exam, right? So all of the changes, the covalent changes that happen to the individual histones, which are then recognized by other proteins. And usually it's a combination of modifications that lead to those. And the code, the histone code, is just which of those particular covalent modifications you have together, not just in one histone, but in the whole constellation that you have in the nucleosome. Histone fold, what's that? Review, yes, there's an exam coming up. Far too soon. The histone folds everything but the tails. So it's those helices in the middle, one long and two short, which then fold together to give you the core structure of all the histones that you have in the nucleosome. So all this, wandering and hand-waving, etc. What's the answer here? Seem to be divided about half and half, relatively speaking, between centromere formation and chromosome segregation. So how do you form a centromere? We haven't really talked very much about this, but we'll talk more about it a little bit later on. How do centromeres actually get formed? How do you get a centromere? It has to do with the sequence of the DNA. It actually really doesn't have to do with the nucleosomes at all. However, the segregation of chromosomes does have to do with that, and that has to do with, again, it's proteins interacting with some part of the nucleosome. In the case of CNPA, it's actually a different histone which is there, but the same thing is true for all of the histone code-reader complexes. It's a different protein that's interacting with the histones. In the case of CNPA, it's all of the microtubules involved in chromosome separation. So it is C, yes. Sorry, I should mention that again for what's in the recording. So our next question here is something that I probably have beaten on far too much already, but about half of the human genome is composed of protein coding genes, repeated sequences, line elements, sign elements, or introns. So we've already got 107. I think that's about as many people as are here. Do you want to stop the voting? No? Take another 15 seconds? Uh-oh. 111. Uh-oh. Okay, so there are a few more here. Either that or people are doing the best kind of election results and voting multiple times. So, okay, 111. We'll see what the max we actually get to. I suspect for the exam there'll be a few more people here as well. So about half of the human genome, of course, is viral, because that's the coolest thing ever. But a lot of those sequences are the repeated sequences, things you have multiple times present in the genome. I don't know, again, how much Dr. Bartlett talked about these, but we will talk about them more when we talk about transposable elements, probably on Friday, maybe on Monday next week. But those are the oops, where's my pointer here? Line elements and sign elements. Come on, where's the pointer? Here we are. Line elements and sign elements. So these are the retrotransposons that together make up about 40% of all of our genome. So these are, people also call them jumping genes that move around. L1 and ALU are some of the most famous of those. Introns, if you think about most of the genes that we have in the genome, where we're going to actually have some protein coding genes, what actually code for proteins? They're not introns, they're exons. Those exons turn out to be about 1.5% of the genome. So the introns are actually almost 10% of the genome. So it gives you an idea how much more there are of those than anything else. And of course, the protein coding genes are really very small. So everyone gets this. This is good. One of the things that I try and do for clicker questions is not have something that you can find immediately from going back to your notes. But this one was unfortunately one of those that could actually, I think, be easily found that way. Okay, so as a quick review, again, hopefully these are the things that Dr. Bartlett talked about. And again, I didn't hear a recording of his lecture. Did somebody make one and post? I didn't see one posted at D2L. But if there is an audio recording, please send that to me and I will post it for everyone else to take a look at. This one, as all the rest of them, should be put on YouTube. Yeah?
I just had a question going back to the whole kinetic core thing.
Yeah.
We have the pretty picture of the centromeres and how there's the CNPA, and then there's that, I guess
Okay, so basically the question, if I'm going to paraphrase here, the really beautiful image that they have in the textbook, the cartoon of what the kinetic core is supposed to look like in terms of interacting with this NPA, that centromere-specific nucleosome. So those are all protein complexes. So it's a whole complex of proteins that sort of interacts with the microtubule. And I think that in cell biology, there's a lot more discussion about exactly what those individual pieces are. If you're interested later on in the textbook, I think it's chapter 20 or 22, they get into a lot more detail about all those other things which are there. So we're mostly just concentrating on the histones now.
We just need to know that it attaches to that protein complex.
Yeah, that it's involved in the kinetic core and that it's that nucleosome, or I say the nucleosomes that have CNPA are going to be those going to interact with those kinetic core proteins. Okay, another quick thing, again, about exams. That person asked me a question about this earlier. I do not go back and look at the textbook when I come up with questions for the exam. I only look at my lecture notes. So and when you're thinking about studying for the exam, don't read through this, how many gazillion pages in the textbook, but look at what's in the lecture notes. Yeah?
That would have been totally helpful, like, first week.
I thought that I mentioned that. It should be in the syllabus, but if not, and again, maybe I missed it earlier on. It's a great textbook. There's really great information there. But it's not all going to be on the exam. It might be on things like MCATs or DATs, but that's a different story. I'm not taking those exams, and I'm not writing them either. So, okay, again, what I think that Dr. Bartlett talked about, I hope he talked about at least a little bit. I know we talked about the 30 nanometer filament already. That's what most of the chromatin looks like in most interphase nuclei. That's where you've got the multiple nucleosomes basically sort of stacked on top of each other. Hopefully, we talked about the fact that not all chromatin is created equal, and that has to do with mostly the fact that some DNA you've got to be able to access, and we'll talk about that for replication in just a couple of minutes. But some of the DNA has to be available for lots of other proteins to interact with, and some of it doesn't. And so a lot of these end up being separate structures that in some cases you can actually see, you can visualize by looking in the microscope, and particularly if you're looking at polytune chromosomes or some of the lamp brush chromosomes, you can literally see loops, and those big loops are the areas of usually active DNA. Those that proteins are reversibly interacting with. Usually that's going to have something to do with transcription, making RNA copies of that particular piece of DNA. Topology, we'll talk a little bit more about. Again, that's the clicker question that Dr. Bartlett had last time that he got from me that I haven't talked about yet. Hopefully we will talk a little bit about supercoiling later on. Again, maybe later today, but much more likely on Friday. Epigenetic inheritance. This is just the very beginnings of epigenetic inheritance. All that epigenetic inheritance means is that you can pass on from mother to daughter some kind of heritable information that's not coded in the DNA sequence. So it can be lots of different things, but hopefully, again, Dr. Bartlett should have talked about the modifications in histones. And so those histone modifications can be passed along through different generations, and usually that has to do with the replication step. So if you replicate your DNA, and then that DNA is associated with particular histone modifications, or for that matter, alternative histones like CNPA, those then are going to reassociate with that piece of DNA. And so the centromere, if we just take that particular example, will stay the centromere and stay associated with those microtubule proteins that we're talking about, because as you replicate those alternative histones, or the modified histones, they're going to stay right there. And then in terms of the new histones and new modifications, the way that those are going to be inherited is that you have a histone reader complex that's going to recognize a particular modification on your histones that's also connected to a histone writer complex that will make more copies of those same modifications. And so once you have one modification, it's going to lead to that same modification on the histones that are near to that particular sequence, which is there. So does that make some sense? No sense? Epigenetic inheritance is really fascinating, and it's something that we're just slowly beginning to understand. There was a really cool paper in Science last week about some of the RNAs that are packaged in sperm that are important for epigenetic inheritance. So stuff that we're learning basically every day about epigenetic inheritance. Heterochromatin versus euchromatin, real chromatin, the euchromatin, heterochromatin, everything else. But basically, heterochromatin is the stuff that's compacted. This is highly compacted. Because it's so compacted, it's going to be really hard for other proteins to get into there, which usually means that's going to be transcriptionally inactive. It's not going to be active because you've compressed the DNA so much with not only your modified histones, but then all the proteins that are associated with those as well. What are those modifications on histones? Probably the most common ones are going to be those on lysines, because they've got these wonderful amine groups hanging out at the ends of their side chains. You can either have them methylated or acetylated, and usually one blocks what's happening with the other. Methylation usually leads to some kind of chromatin compaction, whereas acetylation usually leads to an opening up. That's a rule of thumb that doesn't always completely work, but at least thinking about it from a purely chemical point of view, normally what kind of charges do lysines have? They have positive charges. What do they interact with? The negative charges on your DNA. On the phosphates, non-specific interaction, normally happening with nucleosomes. If you neutralize that charge, is what happens when you acetylate it, then you're going to have less interaction. These things are going to open up. Also, yes, you neutralize the charge with methylation as well. And what that tells you is it's not just those charge-charge interactions, it's the other proteins that are associated with that. So the PhD domain, which interacts with trimethylated lysine at position 9 on histone H3. Sounds like a really specific kind of interaction, but that's true. That's what happens. You have very specific interactions with very specific modifications that happen on the individual histones. Hopefully, alternative histones we covered here already when talking about CEN-PA. Those alternative histones all have the same histone fold. Again, the 3 helices in the middle, because that's the only way they could even assemble into a nucleosome. But we'll have different sequences. Of course, it's going to give you a little bit of a different structure, different things it can interact with outside of that histone fold. Dr. Bartlett said, got to some really cool slides at the end of his lecture. He had no idea what they were. Those were some images which I pulled for some other textbooks. That was basically looking at where you have some of these very specifically modified histones. There were antibodies showing that some of these modifications are very specific, and you can see them in whole chromosomes. They'll be concentrated in particular places. So if you think about where you have trimethylated lysines, usually leading to some kind of inactivation of that part of the chromosome, what's that inactivation usually due to? It's the formation of heterochromatin. And that's the compacted part of the chromatin, which if you look at chromosomes that are otherwise easier to see, like the polytene chromosomes, Lamprecht chromosomes, you can then take an antibody and look at those. You see where you have the heterochromatin. You also have those specifically modified histones. So that was what that was supposed to be showing there. And then in terms of getting a metaphase chromosome, this is the most compacted form of the chromosome. You need cohesin and condensin, which require ATP to really pull all those loops together before you can get the microtubules interacting with the alternative histones, kinetochores, pulling everything apart. So other questions about that lecture before we start talking about replication? how to make more copies of these chromosomes. Okay, good. So our review for today, we'll start talking a little bit about mutations. We'll talk a lot more about mutations when we talk about DNA repair. Then how replication works, semi-conservative replication. Hopefully this is something which is not new to you at all, but it was really new when Watson and Crick came up with their model for the structure in the very first place. And it also raised a couple of problems in terms of how do you copy these two strands when they're so wound up around each other. And in fact, a lot of people at the time, some eminent scientists, some of whom earned Nobel Prizes soon thereafter, thought it was impossible to pull the two strands apart and then make a copy of them. And so we'll look at what those different models were in terms of how DNA could have been being replicated. And then, as some people have called it, the most beautiful experiment in biology, the Meselson-Stahl experiment, as they call it, by, among other people, Frank Stahl, who's emeritus professor just down the road in Eugene. And if you're interested in more details, it's only 250-odd pages that are in here. Welcome to borrow it from my library. Talk a little bit about how that actually works. Turns out that people had discovered, and when I say people, mostly Arthur Kornberg and people working in his lab, had actually discovered the enzymes that did this before Meselson-Stahl did the experiment and showed that it was really semi-conservative replication. So a lot of this stuff was happening really in the late 1950s when all of this was going on. Proofreading, talk a little bit about how proofreading works. Basically, all that proofreading is, you put in the wrong nucleotide, I say you, anthropomorphizing here. The DNA polymerase puts in the wrong nucleotide, how it gets taken back out. And then we'll look a little bit at all the rest of the things that are going on while you're replicating. The DNA polymerase is a wonderful enzyme, does a great job, but it has to have a whole bunch of other proteins that are involved in actually making more DNA. And people call this the replication fork. We'll talk a lot about some of the other proteins that are there. Those include the so-called sliding clamp structure. The sliding clamp is a basically ring around your DNA that forms around the DNA. It's absolutely critical for replication. All organisms have these sliding clamps. Curiously enough, they're different in bacteria, archaea, and eukarya, but they all serve the same role, which is hanging onto the polymerase, making sure the polymerase doesn't fall off. And polymerases are really good at falling off of DNA, so you absolutely require these sliding clamps. All this together comes into the replisome, and then we may get around to talking about mismatch repair. What happens with mismatch repair is basically when proofreading doesn't happen or it's incorrect, then you need mismatch repair to deal with that particular problem. And then, again, if we have a chance, we'll talk about supercoiling. It's one of the ways that you can compact DNA. It's really important for compacting DNA, but it turns out that you make a bunch of supercoils while you're replicating, and that causes a big problem. And so you have to deal with supercoiling while you're undergoing replication. Look at some of the differences between eukaryotes and bacteria, and then talk about some origins. Let's start very briefly talking about mutations. Mutations are what happens to DNA which changes the genetic information which is present there. Turns out that, as I may have mentioned before, we already talked about why DNA is a horrible molecule for being the genetic material because it's just so darn big. You've got to have 2 meters of DNA that you compact into the nucleus of each of your individual cells. So that's one of the problems that DNA has. The other problem, or one of the other problems that DNA has, is incredibly susceptible to chemical changes that happen to all of the bases, happen to the sugars, happen to the phosphates, etc. We'll talk more about those when we talk about, again, DNA repair, but that's after the midterm, so who cares at this point. But when you do have some kind of chemical change which changes the genetic information which is here, that is going to be change in that genetic material which is there. People have looked at how often this happens. Turns out it's very, very infrequent. It's about 1 in a billion in E. coli, and actually turns out in humans as well. So per replication cycle, so you go through, copy 1 billion bases of DNA, be you E. coli, be you Sulfolobus sulfatericus, my favorite organism, archaea, or in humans, about 1 in a billion of those copies you'll put in the wrong nucleotide, and it will stay there. And so means that it's past proofreading, it's past mismatch repair. And what that means, you can do some back-of-the-envelope calculations that any organism, if it's got this mutation rate, and since it seems to be mutation rate, we've got an E. coli in humans, in archaea, that's probably about where you're at. What it means is you can only have about 50,000 proteins that are essential proteins. And the reason for that is if you have multiple generations, each time you copy a genome, have this many changes that happen, you could only maintain about, again, it depends, 30 to 50,000 proteins, and have those as be really essential. Because otherwise, if you had more proteins that were essential, you'd make too many mutations. And then those would end up being deleterious. Because most mutations are bad. Probably because we've had billions of years of selection, any of those individual mutations change in your genetic material. In some cases, again, relative to the genetic code, that's going to change your protein. That's going to be a problem. And any change that you do to your protein, change the protein, change the structure, change the function. Sometimes that'll work. But again, most of the times it ends up being pretty negative. And we know that that's the case because cancer is a really good example of how we have lots of mutations that end up causing lots of problems. We saw when we looked at the karyotypes. You have lots of mixing and matching of chromosomes that don't get repaired, but also if you look at the individual mutation rate, the number of individual nucleotides that have changed in cancers, those also get way out of control. So yeah, a lot of times you have mutations that are bad. However, if you had no mutations whatsoever, that sounds like a great thing, right? Absolutely no mutations, wouldn't that be wonderful? The genetic material's maintained forever exactly the same. Well, then we would all probably still be slime, squiggling around in some hot spring or other. So basically, the fact that you do have mutations and that a very, very, very small number of those turn out to be beneficial, and beneficial again looking at it from our point of view or from the yeast point of view or from the archaebacterial point of view, that allows then evolution to take place. Because you have to have some basis, some kind of diversity in order to have selection. And so, yeah, DNA may be a really crummy molecule because you can get all of this damage, but it probably is the genetic material that all cellular organisms use because there's a small amount of change that happens to it. And that change is what can happen and allow evolution to take place through that. So let's talk about how most of the time you do a really good job, and you being you and all the bacteria in your gut and all the archaea in the hot springs do a great job of replicating. So this is, in fact, what Watson and Crick predicted very soon after their original paper. And yes, I say I always have exam questions from my lectures. There's one exception to that, and that's the question that will be on Watson and Crick's paper from '53 that everyone's read because it's on D2L and I told you you should. I'm sure you will by the midterm. This was the prediction that you would have the two strands come apart, and then once you have those two strands come apart, then of course you can make copies of each of those strands because you've got the redundant information. Copy those, you end up with redundant information as well. So their prediction was that you would have these two strands that were wrapped around each other, pull those two strands apart, and copy each one of those strands. So here, this is an image from a different textbook, but I liked it partly because it was actually wrong. They put the 5' and 3' in incorrectly. But here we have the 5' end. Just follow this around. down here to the 3' end in red. This is the old one. Always anti-parallel, so the 3' end, you follow it around down here. Here's your 5' end of the old, and you pulled these two apart. It's great. You can copy them. Here goes up from 3' to 5', and here goes from 5' to 3'. Unfortunately, no one's actually found anything that goes from 5' to 3'. So, great idea. Ended up being a little bit more problematic later on. But this was the general concept that Watson and Crick had. And so basically, this is the so-called semi-conservative replication process. And we'll talk about the conservative replication in just a second, just so you get the idea what they mean by semi-conservative. All that semi-conservative means you hang on to 1/2 each time when you do your replication. And so that's shown here. It's a little hard to see color-wise, but the orange are the old strands. The red ones here, I guess, are the new ones. So after the first generation, you'll end up with 2 strands, which are a mixture of new and old. After the second generation, you'll still have 2, which are a mixture of new and old, but the new strand is going to get copied. So you have multiple new strands, and so on and so forth through multiple generations. But you're always going to have this mixture of one of these original strands that you started with. So that's a semi-conservative process. At the same time, again, because I mentioned earlier, people had trouble thinking about how you could pull those 2 strands apart. There was a whole another group of people who thought that there was conservative replication. And all the conservative replication says is that you actually keep those 2 strands together the whole time. And you just make a copy from that. Now, what would that copy look like? Here, after the first generation, you would have double-stranded copy of what you had started with. And at first, okay, how the heck can this work? But we talked about, when we talked about the DNA structure, that all the information about the base pairs is actually present in the major groove of DNA. So if you had some adapter which could say, okay, this is the sequence that we have here of those base pairs, you could then say over here, when you're making a copy of it, yeah, we know what the sequence is over here. We can make that sequence over here. So that would be the conservative replication process. And again, a lot of people thought that this made sense because you didn't have to pull those strands apart. You could keep the strands together. Much more stable kind of interaction. But here, instead of having mixtures of the 2 strands, now old and new, you're going to have 2 strands that are old, 2 strands that are new. In the next generation, you'll have 2 strands that are old, and then 3 copies of 2 new strands. So those are 2 different models. A third model is what's called dispersive replication. And this, it turns out, is how almost all DNA repair works. Certainly if you have some kind of point mutation. And what happens is it's a dispersive process. And all that this is saying that here, instead of pulling the strands apart completely and making a copy, keeping one strand all the same, is you just mix and match. You'd have different pieces. And so you copy part of this strand here is going to be new, this strand over here is going to be old. So it's just a mix and match process. You cut out pieces and move them around. Hard to imagine how this actually would be working at the whole chromosome level, but again, this is really what happens with most DNA repair machinery, is that you take these little pieces and copy little pieces. What you see here is if you're doing that and follow through with this first generation is going to be half new, half old. The next generation is going to be 1/4 old and 3/4 new. And then the next generation will be 1/8 old and 7/8 new, and so on and so forth. So the question came up with these 3 models, basically which were around in the mid-1950s, right after Watson and Crick had come up with their model, is how to differentiate between all of these things. So there were a number of different labs that were working on this at the time, including a guy who basically said that Meselson and Stahl came up with the best experiment in biology, fellow by the name of Gunther Stent, who also tried to do exactly the same thing and failed. But what Meselson and Stahl came up with was really a great combination of two people with different expertises. So Frank Stahl was really, really good at growing bacteria, and growing bacteria in a way that was very repeatable. And that turns out to be absolutely critical for doing these kinds of experiments. At the same time, Matt Meselson had basically discovered how to use the ultracentrifuge with specific heavy ions in it and do what's called equilibrium ultracentrifugation. And so what equilibrium ultracentrifugation does, and we'll talk about this again right at the end of the course, is it separates whatever you put into the ultracentrifuge based on density. And so just looking at the absolute density of what you have in your particular sample. And so what Meselson and Stahl came up with was this idea of taking E. coli, which Stahl was really good at growing, and grow it first with heavy nucleotides. And so what they decided to use for heavy nucleotides here were nucleotides that were labeled with N15. So why N15? That's a little hard to tell back there now, but when you come back, take a quick look at the structure a little bit later on, the N's in this model are all the blue things. And if you think about the base pair structure, particularly the bases, there's a whole bunch of nitrogen atoms in them. In fact, way more nitrogen atoms than anything except for your carbon and hydrogen. Really hard to get a whole bunch of deuterium at the time, and the carbon isotopes were also a bit of a drag in terms of getting a hold of. So lots and lots of nitrogen which is present in your DNA. And then, since this was the late 1950s, all the people on the Manhattan Project had generated these different isotopes. You could actually get the precursor to all of your nucleotides. In this case was ammonium chloride that was specifically N15 labeled or specifically unlabeled, so mostly N14. So you grow your E. coli in the N15. All of your E. coli DNA is going to have N15 in it. Then you switch it to growing in N14. All of the new DNA which is being made will be light DNA in the process. So once you've done that, now you need to separate the DNA from the cells, which actually was also not a straightforward process to do at the time, and then use Matt Meselson's equilibrium analytical ultracentrifugation to just separate based on density. So this is what that process looked like. If you just took the DNA from the E. coli that was grown on the heavy DNA, it would end up here down towards the bottom of your centrifuge tube because it's relatively high density. Then, after growing it in this light N14, there are really three possibilities that could happen. Remember, there are three models: conservative, semi-conservative, and dispersive in terms of your possible results. So here, and this is, I think, partly why people called this the most beautiful experiment in biology, is because you could make really clear predictions what the results were going to be before you even did the experiment. So if you had semi-conservative replication, you would expect to, after the first generation, you remember we're going to have now a mixture in your double strands of heavy and light. So you see something in between heavy and light, which is here, this what's called the hybrid band on the gel here. In your ultracentrifuge. So that's going to migrate a little less than the dense DNA. And after the second generation, you'll start to see some DNA which is all N14 DNA. And the longer you grow this, you'll have more and more of this band, but you'll always have some of this N14, N15, one strand each. Because you remember with semi-conservative, you always are going to keep one of those strands, which is there, always one of the old strands. On the other hand, if you have conservative replication, which is what's shown down here, you'll have always heavy because those strands stay together, and then an identical copy of the two strands which are light. If you have the dispersive replication, after the first generation, this actually looks identical between dispersive and semi-conservative. It's half and half. So in this case, you have to go to another generation where now instead of being half and half with your dispersive, it would be 1/4, 3/4. And then after the third generation, 1/8, 7/8. And so it's all going to be moving together as opposed to seeing the two bands that you have here to start with. So how was this experiment done? This is Matt Meselson in front of what they called the big machine, the Model E analytical ultracentrifuge. I think there were 4 of them in the world at the time. This particular one was at Caltech. This whole big machine here basically took up about a whole wall and spun this rotor in it, which had a window in one side that UV light was shone through onto a photographic plate down here at the bottom. What did this give? This is one of the figures from their paper. If you look at just the DNA that you start with, again, shining UV light at this, we talked about UV absorbance of DNA. That's lower for double-stranded DNA, higher for single-stranded DNA. They're just looking at here the double-stranded DNA. As you grow the E. coli, this band starts to move upwards, and after one generation, it's clearly moved from down at the bottom to up here. What does that mean in terms of the 3 models? Which one has been ruled out by that process, just up to the first generation? Conservative is ruled out. Now, if you keep going up to about 2 generations, now you start to see 2 bands, and after 4 generations, you can still see this hybrid band here, which is somewhat less dense than what you started with. But most of this is here. So this was really clear evidence that supported only one of the 3 models for replication. So again, 1958. So only about 5 years after Watson and Crick had come up with their model for DNA, Meselson-Stahl had really nice experimental evidence for that being actually a semi-conservative replication model. So what was doing that? What was the process that was doing this? Turns out that Arthur Kornberg actually had purified an enzyme before they published their paper in 57 called DNA polymerase. DNA polymerase 1, because it was the first DNA polymerase that he discovered, studying in E. coli. And what he found is this was an enzyme that would take deoxynucleotide triphosphates if you had a template and a free 3' OH and make these individual triphosphates into a bigger piece, in this case a DNA, a double-stranded DNA molecule. But as I say, this particular enzyme needed both a template and a free 3' OH, which is usually what people will call a primer. So you had to have a DNA strand to copy, which again, through the semi-conservative process, but then you also had to have a 3' OH, and that 3' OH would serve as a primer where you then add each of these individual triphosphates. Triphosphate, as we tried to show in the lecture where my picture disappeared, you have hydrolysis of this triphosphate, giving you a pyrophosphate that then gets split into the individual phosphate molecules that hooks your new nucleotide in due to the base pairing interaction. So you give you the right one. That gives you another 3' OH this 3' OH can be added to by the next triphosphate, and so on and so forth. So you can extend the strand by the presence of this DNA polymerase. Many years later, people solved the structure of these DNA polymerases, and people call these sort of hands, or a right-handed structure. So I'll try and bring my hand up over here. The idea is that you've got basically a thumb sticking up. That's supposed to be this structure here at the top. Your fingers here that basically grab the DNA, and it's down in the palm of the hand where you actually have the activity, which is the extension of these 3' OHs. So that's the activity of this DNA polymerase, and more specifically, we should be calling this a DNA-dependent DNA polymerase, because it has to have a DNA template, also has to have this primer process. So one of the things you'll notice here is that it's adding always to the 3' end. And so people will talk about this. You're continuing here this process. You start at the 5' end, and you're going to the 3' end. So the polymerization process here starts at 5' and goes to 3'. People almost always talk about this as a 5' to 3' polymerase. It's just another way of looking at that activity. Base pairs happen here between the base. You have hydrolysis of the pyrophosphate. This phosphate gets attached to the 3' OH, generates another 3' OH, and you move on through the whole process. We talked about this hand structure. Here it's even more nicely shown as a hand. This is the actual model for the structure here. But most important as far as this is concerned is that we have a couple of metal ions that are present in the active site. When we talked about active site, when we talked about enzymes, this is the incredibly complementary structure. So we've got the individual amino acid side chains. Most of your protein is just there in order to pull the appropriate amino acid side chains into the right place. Once you've pulled those amino acid side chains into the right place, then you can bind very specifically to your substrate and allow the reactions to take place. Many of these enzymes also will have metals which are present in there. I also like this particular model because it shows that your template strand here is really kind of bent away from the active site of the structure. And that active site also, and we'll see this when we talk about proofreading in just a second, the active site here is really very much about structure. It's not about base pair interactions. So again, how do you get the right nucleotide added? It's because it base pairs to the nucleotide on the opposite strand. But if you have a DNA polymerase, which is going to be checking to make sure that, okay, I've got a C here, I better be putting in a G. I've got a T here, I better be putting in an A. I've got an A here, I better be putting in a T. That's going to be a lot harder than saying, oh, have we got the right structure of the bases which are there? And we see that's really important. You've got a kinked template because the template is going to be out of the way and only brought back into the right structure when you have those right base pairs that form. And we'll see that again in just a couple of slides here when we talk about proofreading, what happens when it goes wrong. So I mentioned before, what Kornberg found was this 5' to 3' polymerase. And rumor has it that Arthur Kornberg kept telling his students, well, okay, we found the polymerase that replicates one strand. We need to find the polymerase that replicates the other strand, right? Because Watson and Crick said you pull the two strands apart, right? And then you replicate them. Well, if you pull the two strands apart, one of those is going to have a 3' end, so you can go from 5' to 3' on the opposite strand. The other one is going to be flipped around. So now you need a polymerase that goes from 3' to 5'. No one had been able to find one of these polymerases. So what's going on with a replication fork? Now, one thing that was found again pretty soon thereafter was first in just looking at DNA. here, electron micrograph, this is a DNA which is replicating. There were clearly these loops that were forming, and this is where DNA replication was taking place. So people also call these replication forks, just a two forks here. So if you blow up one of these replication forks, you end up with your old DNA and then the new DNA which is being made. 5' to 3', it's easy. Kornberg found this polymerase. On the other hand, how do you make all these pieces on the opposite strand? Reiji and his wife, Okazaki, I'm sorry, forgetting her name now, found these small pieces of DNA which were made in this replication process. And eventually what they found was that these were these short pieces of DNA which were all being made also from 5' to 3', but little short pieces. And all these little short pieces are also now known as Okazaki fragments. Fortunately, they were looking in bacteria where the Okazaki fragments are relatively big. They're about 1,000 nucleotides. In eukaryotes, they're actually relatively small, these 100 nucleotide fragments. But what this means is that you have multiple pieces here on what's called the lagging strand and only one new piece of DNA on the leading strand. What this means is that people will talk about this as being a discontinuous replication process because we've got these individual pieces which are being made here. But these individual pieces are being made discontinuously. This piece, on the other hand, is a continuous process. So if you want to talk about how DNA replication works, it's semi-conservative and semi-discontinuous. So semi-discontinuous, semi-conservative replication is how replication is actually working here. Semi-continuous because we've got some pieces that are made all as one, some pieces that are made individually, and then semi-conservative because you hang on to one each of those strands. So far, so good? Yes. Okay, so now let's talk about how we make few mistakes. A couple of different ways that we've evolved, we and E. coli have evolved, to make very few mistakes, how you get the addition. Now remember I talked about the kinked strands already, the kinked template strand, and that you can see up here in this model where this is the kink on the template strand. As I mentioned before, the polymerase itself doesn't know which base it's putting in. But what it does know is that the base forms a pair. And the way that happens really has to do with not so much that A's and T's and G's and C's are matching with each other. It's that they form this nice flat structure. And so it's a nice flat structure. You only get a nice flat structure if you have the right base pairs that form A's, T's, or G's, C's. If you have AA's, if you have TT, GG, CC, any of the mismatches don't give you a nice flat structure that you have here in your DNA. And so it's really a base pair which is being detected as opposed to the right or the correct base which is coming into your structure. So the way that's done is by basically looking at, is it a nice flat structure? How does that happen? It's a helix, an alpha helix, which is present in the DNA polymerase, which basically folds down on top and says, hey, is this a nice flat nucleotide here, or is it not? And if it's not, then you're not going to have this nice fold, which is going to bring the amino acids that you need to have the addition of the individual nucleotide. So if you don't have a good base pair here, this is not going to stack properly. You're not going to bring the individual side chains in order to catalyze this base reaction. So it's all about the structure of the base pair and the changing in the structure of the DNA polymerase, which gives you the appropriate base pair which is going to form. Yeah?
Sorry, I missed it, but the spiral that goes on, what is that part of that detects the flat structure?
Oh, the green spiral up here? This one?
The thing that's doing the detection, is that part of the DNA or is that part of the polymerase?
Okay, so this green thing here is an alpha helix. It's part of the polymerase.
Okay.
And it's actually shown down here as being this part right here. And so this is the polymerase. This is a part of the polymerase. All the rest of it has been taken away. And again, it's like what we talked about protein folding before. Most of the amino acids in your protein are just there to get these very few amino acids into the right place. And so that particular case is the right place in the alpha helix here. Yeah?
So does it just try each base until it finds a flat one, or is it more precise than that?
So the question is, does it just try and with all the individual nucleotides that could be coming in until it gets a flat one that matches? That's what seems to actually happen. So it seems to be screening through all of the other nucleotides which are there, which seems to be a rather inefficient process, but means that you've got lots of nucleotides around. You've got to have lots of these nucleotide triphosphates around. So yes, it is. It's basically, wait till we get the right one. The right one's there, boom. Okay, we move. Wait to get the right one. Right one's there, you move. And so one of the ways that you can slow down DNA polymerization is by reducing the amount of one of the particular nucleotides. People use that in the lab all the time when they're studying this. Yeah?
How does this cause errors? How do the errors get through this process?
How do the errors get through this process? We're just about to get there in just a second. So part of the process is that it's going very quickly, and we didn't talk about that, but it's literally hundreds of nucleotides a second that you're moving along. In the case of bacterial polymerase, it's even faster than that. We talked a little bit when we talked about the structure model that Watson and Crick came up with, the keto versus enol. And so there is actually tautomerization that happens. If you have the wrong tautomer, which is there, when you just happen to be screening through all these nucleotides, that'll actually give you the right base pairing, the right conformation, but that tautomer is going to switch and they'll give you the wrong conformation a little bit later on. So this is not 100% efficient. In fact, it does make a number of mistakes. How do you deal with those mistakes? Well, the first one is, again, it's a structure detection. You detect that the base pair is wrong. If the base pair is wrong, then you want to get rid of the last base which you had in there. Again, it's the configuration of the base pair which is noticed. It's not the individual base which is there. So polymerase makes a mistake. It's trying to see, is it flat, is it flat, is it flat? It's flat enough. Okay, good, we'll actually make this one. Some cases will be wrong, and again, very often it's the tautomer of the nucleotide which is there. Smaller percentage, but it's still there. So the wrong base gets added. Then there's an exonuclease which will chop off that incorrectly added base. That exonuclease backs up, so it's a 3' to 5' exonuclease because you just added on the 3' end. Okay, so your exonuclease has to go back from 5 to 3, generate a 3' OH that can then be added to by the polymerase. All of the replicative polymerases have a separate domain which has this exonuclease activity. Just a quick reminder, exo means coming in at the end, endo means cutting in the middle. Exo is going to be chewing at the ends of your nucleic acid, endo is going to cut in the middle. So the exonuclease will chop out that individual nucleotide and you will restart your replication because you've generated a 3' OH. This only works with 5' to 3' polymerization. Now, why is that? That's basically shown here in this cartoon. So this is the way it normally works. You have your polymerization. Here's your nucleotide triphosphate. Cut off your pyrophosphate. Add this. You've got a 3' OH. You do proofreading. You chop off that last little piece. You've got a 3' OH. You've got your nucleotide triphosphate. You can extend here normally. If, however, you had a polymerase that's going from 3' to 5', now you'd have your triphosphate at this end because you're always going to have the same linkage. We always know we've got a 3' and 5' linkage. Here we have a hydrolysis of pyrophosphate. Add on your nucleotide. Here's a nucleotide. You can continue here because you've got pyrophosphate. Put on another nucleotide. However, if this were the incorrect nucleotide, you don't have the right structure of that base pair, it would get cleaved off. That cleavage now would leave you with a 5' monophosphate. That 5' monophosphate you can't get a pyrophosphate from to continue this process. So you can't have 5' extension and still do proofreading with the same process. Whereas if you have 3' extension and you're using the pyrophosphate from the incoming nucleotide as opposed to the pyrophosphate that you have on the end of your chain, now you can do proofreading. And this is probably why all of the DNA polymerases, and for that matter RNA polymerases that have been found to date, all have this 5' to 3' polymerization. None of them go from 3' to 5', because if you went from 3' to 5', you couldn't do proofreading. This is a review of what happens here. Normal polymerization process, which we have over here. Again, the hand structure down in the palm. You're checking to make sure that this has the right conformation, the right kind of base stacking information. If it's wrong, then this extra domain comes in here. It chops off that individual nucleotide, and you can restart the whole process. Through this process, the polymerization process, you're asking about, again, random kinds of interactions here. About 1 in 10,000 nucleotides get put in incorrectly there. So 1 in 10,000, okay, that's not very many, but it's a heck of a lot less than the 1 in a million or 1 in 10 million, which is what we actually see. So of those 1 in 10,000 that get put in incorrectly, 99 out of 100 get taken care of by this proofreading mechanism. So the backing up the 5' to 3', 3' to 5' exonuclease activity that chops it off. Then, and we'll talk about this next time, you have mismatch repair, which is also about another 1 in 1,000. You can add all these up. It ends up in 1 in a million or 1 in 10 million, which still escapes that whole process. And that's the whole replication process as it moves forward. Last thing that I wanted to talk about here is some of the other proteins that you have at the replication fork. All of you have been listening carefully, have noticed that I mentioned all the DNA polymerases have to have a template. Okay, you have to get that template somehow. You've got double-stranded DNA that has to become single-stranded DNA. But much more importantly, you also have to have an OH, because all DNA polymerases extend 3' OHs. So they add a nucleotide triphosphate to something that already has a 3' OH. Where does that 3' OH come from when you start out? Not such a big deal if you're talking about the leading strand, because that starts once and goes in. But if you're talking about the lagging strand, you need to start a whole bunch of times because it's a semi-discontinuous process. So where do you get that from? Turns out it's an RNA polymerase. So RNA polymerases can add nucleotides without a primer, whereas DNA polymerases need to have a 3' OH to extend. So that's the DNA primase. DNA primase is actually really an RNA polymerase, which takes RNAs, adds those RNAs, puts a couple of these together, and then the DNA polymerase will find this OH and extend it. Turns out that this RNA polymerase, like all RNA polymerases, doesn't do as good a job at making sure that all your base pairs are correct. So it's a relatively low fidelity. It puts in a lot of mistakes. So you need to get rid of those a little bit later on. How you get rid of those are these things called RNase H. RNase H is an RNase. Gee, surprise, surprise. What does it do? It chops up RNA. But RNase H means that it cuts up RNA, which is in a DNA-RNA hybrid. So when you've got a DNA and an RNA together, that's where RNase H is going to work. And then because of these Okazaki fragments, there are all these individual pieces that have to be hooked together. And how do you hook these together? It's a protein called DNA ligase. There are other processes that are involved here, but we'll talk about those on Friday.